So, ladies and gentlemen, our special session, speaking on the DNA of the future, we have the pioneering genomic researcher, Dr. Craig Venter, and as chairperson, we have with us Professor Samir Ramachari, Director General of the Council of Scientific and Industrial Research. Professor has pioneered functional genomics research in India and led the Indian Genome Variation Consortium project, and is presently coordinating a national network project in silico biology for drug target development. Professor, please introduce Dr. Craig Venter.
Thank you, India Today Conclave, and it's a great opportunity and honor to chair Dr. Craig Venter, second time after 2003 at Ashwacham and now at this enclave. Dr. Venter, as you have seen already in the news items on the book, I won't read it out. Instead of that, I just show you a few slides. Where do I show? Those who know nothing, the human genome is a 3 billion nucleotide sequence, base pairs, and if you start reading them, ATGC, you might take about several years to finish reading them. But this is what, in the history of 50 years of DNA, if you look at the first half till 1991, there was a series of people have participated and developed what it is. But since 91, you can see the first ESD sequence, the first genome sequence, the first together, the open, that is the Human Genome Project, and the cellular genomics, Craig Venter's work, and that was the first phase of genomics. Subsequently, Dr. Craig Venter didn't stop. He moved on in 2004, did ocean metagenomics. Then in 2007, the only individual whose full human genome sequence is available today, you have it here in this auditorium, is that Professor Craig Venter. And 2008, synthetic biology for artificial life form, and synthetic biology will create a new life form, and from this, the entire industrial transformation will take place. I'm sure he is going to take you today. He has not only done this, he has created an X Prize Foundation to see all of us can get our sequence done in 40,000 bucks, 50,000 rupees, and in a day. And this foundation is put up this challenge for the future. While world was sequencing and we decided that this sequence will be available, at India we just created this network of capability to use functionally. We have the large hospitals and huge number of patients. We created the largest DNA material from complex disorders like neurological, psychiatric and all, asthma, diabetes. And we first a country that whose entire genetic profile is just got mapped in 55 populations across India, and you can see the parts of India which goes with hap map of Chinese, Japanese, some with the Caucasian, European, and Dravidian and Austroasiatic individuals. This map clearly shows that the entire India continues from the entire world map from the end of this, and that's the Southeast Asia. It covers the entire from Caucasian to the Japanese. India is a melting pot and ideal for clinical trials. And this is the risk disease profiles. You can see the regions of red are the protected for diabetes, protected for HIV, and the high risk of hypertension. So depending on where you belong, your risk is that. This is the first map of any country with respect to a single drug response called asthma drug response for salbutamol. And where the red ones are the hotspot of poor responders, and other ones are, so if you are there, you need steroid. What it made difference, what did Craig Venter do differently? When everybody is interested in science to do things that are doable, he got into doing science which are not easily doable. That means no technology exists, no knowledge exists. And imagine in 2003 he proposed to design microbes to become an industrial worker, to create hydrogen fuel, capture carbon dioxide much before the global warming, and to create energy, sugar, glucose. Same time, why I say this, because when developing predictive medicine, 1 to 3, you can see it was high-risk area. There's no clue of market. Today it has reached a region where you have market demand exists. So it is our interest, knowing him, knowing such great people. I'm very fortunate that we initiated 10 years back, and we are there with all these diagnostics markers. And it is important that ahead of time and future vision is important. Synthetic biology is a design, a fabrication of biological systems that do not exist. It creates controversy, it creates a lot of discussion, and you will listen to him how he has gone about in creating this mycoplasma laboratory. You have a name here. You have a name, no? And he will discuss about this. And he has created this, and this is the first organism which hasn't come out of Darwinian evolution. It needs a lot of science in it. It is not simple. And we need an integrated science, and that's what it needs. It can do many things. It can do bioenergy, bioplastic, biosensors, drug delivery, new therapeutics, you name it. And it can replace the entire chemical industry in 20th century. I thought this single statement of Craig defines that we are not individual collection of genes, but what we are with the environment, that individual, what we are, and happen to be we are 6 billion individuals who are distinctly different than each other. I just thought in 2003, April, when he came for associate, we visited our center, I played this clip, and you can see the vision from the visionary's voice. Can you play the sound bite, please? What happened? This one. No.
Is a field that has applications to every part of human society. We have an institute that's working on biological energy, trying to capture CO2 out of the atmosphere, convert the CO2 into useful products by capturing the carbon. We're trying to engineer the photosynthetic process to drive hydrogen production in a very efficient form directly from sunlight.
So I invite no other than Craig Venter to address this audience. Craig.
Well, thank you for the extremely kind introduction. Can we switch the slides? Oh, thank you. But it's nice to be back here again. 2003 seems like a very long time ago now, but a lot has happened since then. So let me update you on the rapid changes in genomics over a short period of time. As you heard, starting back in 1995, we sequenced the first genome of a living organism. This was a computationally intense project, and we used computers instead of large numbers of humans to do this work. We scaled up very rapidly, so only 5 years later, we were able to do the human genome in 9 months, going from 1.8 million letters of genetic code to over 3 billion. It turns out the genomes that were published in 2001 were not complete or accurate or even representing what we thought about humanity. So the message out of 2001 is that we all differed from each other only in 1 letter out of 1,000 of our genetic code, and all humans have the same genes. Now, we just last September published the first complete human genome containing the sets of chromosomes from both parents. And we found, in fact, that humans differed very dramatically from each other. We're 1 to 3% different from each other across this planet instead of 1 letter out of 1,000, and we don't all have the same genes. In fact, each one of you at each loci on your chromosome have 2 very different genes, or in some cases only 1 copy or even no copies of genes. There's a gene associated with detoxifying environmental toxins that only 1/3 of the Caucasian population has 2 copies. Another 1/3 has 1 copy, and 1/3 has no copies at all. We see this now across the genome and across the genetic code. So we're just now beginning to look at the human genetic code accurately for the first time. And we're trying to scale up, whether it's through the funding $10 million prize from the X Prize Foundation, whoever comes up with technology to sequence a genome for about $1,000, so that it can be truly universally applied. But what we need to do is capture now phenotypic information. Think about all the information that would describe each one of you, physically, mentally, historically, medically, and thinking of digitizing that information so we can compare it to the human genetic code to find out for the first time in history absolutely what's nature and what's nurture. In fact, as the quote that you used shows, that we in fact are also very dependent on our environment. Our environment is in fact quite local. So these new techniques that we establish to look at the environment, we can look at the environment in the human gut or in the mouth or on the skin. And what we found was quite stunning. We literally have millions and millions of genes associated with organisms that live with us on a daily basis. So just think, in your mouth you have over 1,000 different species there all the time. Think about that when you kiss somebody later. The intestinal tract, over 1,000 species. And just imagine what happens when you take massive doses of antibiotics. You change your own repertoire quite substantially. Now you may ask, what role do these play? Well, we know in the human genome we have around 23,000 genes. Those make a large number of proteins, but those proteins can only make around 2,400 different chemical compounds. And so we can start to look in the bloodstream of humans under various conditions, and there's a company called Metabolomics in North Carolina that's doing this. And one of the stunning surprises after any sort of meal, there's maybe about 500 different chemicals circulating in the bloodstream. About 60% of those are what we make as humans. About 30% come from what you just ate. So the wide range of species, it doesn't matter whether it's plants or animals, the chemicals are extracted from those and end up in our bloodstream. But 10% or so are not from what we ate. They're from the metabolism that our bacteria in our guts do with those chemicals. So some people argue you are what you eat. I'll argue you're also what you feed your bacteria and what they feed us. So we are in a very complex milieu of organisms. Very few of these ever cause disease. Only a tiny fraction of all microorganisms and viruses care about humans in a disease fashion. So to understand human physiology, to understand medicine, we have to understand this increasing degree of complexity. We use the same tools to start to look at the broader environment, trying to understand who we shared our planet with. And it turns out if you go out to any nearby sea, each milliliter of seawater will have about 1 million bacteria and over 10 million viruses in it. So I swim in the ocean all the time. Sometimes when I'm out surfing, I swallow a mouthful of seawater, and I imagine the millions of organisms that I just consumed. But most of these were unknown. Our laboratory techniques have changed very little until recently, since the days of Pasteur. People looked to see what would grow in cultures in the lab and decided there was very little biology in the oceans. We in fact did a different experiment. We did a shotgun sequence into the ocean the same way we did for the human genome. And we found just in 200 liters of seawater about 40,000 new species, over 1 million new genes. We then started the Sorcerer II expedition. This was a publication, a special issue of PLOS Biology, the online public access journal, where we described our sailing around the world and taking samples every 200 miles. The publication that was described only covers from Halifax, Nova Scotia down through the Galapagos, the blue dots that you see on this slide. Since then, we have sailed across the Pacific Ocean, around Australia, then across the Indian Ocean, around the tip of South Africa, and then back to Washington. And as a result, now we have a database of over 20 million new genes, about 4 times the size of all the data collected by all scientists using other methods. One of the findings was every 200 miles, 85% of the organisms were unique. So we can actually tell from a DNA sample in the ocean where that came from in the world. These are unique environments, so many people think the ocean just mixes and it's a homogeneous soup. In fact, it's tens of millions of dynamically changing microenvironments depending on the sunlight, the salinity, the pollution, etc. One of the big findings we had was every organism in the top parts of the ocean has a photoreceptor almost identical to the visual pigments in your eyes. Only instead of seeing these organisms use these receptors to capture energy from the sun, not through photosynthesis, but a totally new mechanism. We could track these around the world, and we found that they had unique localizations. So the organisms that could see only blue light exist in deep blue water. When you get to coastal waters where there's a lot of chlorophyll, the photoreceptors see only green light, and that provides tremendous selection for the different organisms just from one amino acid change based on a single letter change in the genetic code. A Swedish group last year, in fact, showed that these organisms can live off sunlight directly, transporting ions across their membranes. So when we think about carbon capture in the ocean, most people thought it was dependent on photosynthesis. Now we know we have this entirely new mechanism that we can think of new ways to harness these novel photoreceptors. The other thing we found is life was far more diverse by orders of magnitude than anybody imagined. There's an organism that was called SAR-11 that was supposed to be a lonely bacteria in the ocean. It turns out there's maybe thousands of related organisms with very distant sequence homologies. Yet from the tools that scientists were using, they thought this was just a single organism. We can look at this data in new ways. We can compare it by the site for collection. We can compare oceans. The bottom is perhaps the most interesting thing. The switch between blue and green light of these photoreceptors has happened 4 times in recent evolutionary history just from these random mutations. But you can ask any question of data, whether it's dependent on changes in temperature, pollution, carbon dioxide, etc. Numerous questions have come out of these discoveries. Were we making new gene discoveries, or was it like the photoreceptors? We're discovering new family members. And if we're making new discoveries, what's the rate of discovery? Can we put this in context? So we assembled all the data from public databases with our ocean sampling data. It took over 1 million CPU hours of compute time, and we came up with about 50,000 major gene families. But every time we take a new sample from the ocean, from the earth, from the air, we discover new gene families in a linear fashion. So we're still in the earliest stages of discovering life on our own planet. This can lead to big questions, such as what is life? And as science is proceeding in a reductionist fashion, can we pare it down to its most basic components? More importantly, for the future, can we digitize life? And if we can digitize it, can we regenerate it out of that digital world? Well, all of DNA sequencing has been the digitization of biology, whether it's from the sequence of bacteria or from the human genome. We're going from this analog world of biology that we live in to the digital world of the computer. Now we're working on trying to go in the other direction, starting with the digital world and going back into the analog world. This is the genome map of Mycoplasma genitalium. It's the smallest genome of a self-replicating organisms. There's other bacteria with smaller genomes, but they're totally dependent on a host organism to be able to live. We just asked simple questions. In fact, that's how most of our science has proceeded. This species has only about 500 genes. The first genome we discovered had about 1,800. And we simply asked the question, is there a smaller genome that's compatible with independent self-replicating life? So we tried knocking out genes using tools of modern genomics to see if we could pare down the genome. But you can only knock out genes one at a time, and doing this we knocked out about 100 genes. But when we look at the metabolic map of the cell, we find out if we knocked out all those 100 genes simultaneously, it's very doubtful that it would lead to a living organism. We decided then at that time the only way to proceed was to make the chromosome chemically in the laboratory where we could vary the gene content. Again, a series of questions. Could the chemistry support making large DNA molecules? And even if we could make this large inert molecule, is there any way to actually activate it in living systems? So DNA sequencing, or the digitation of biology, has been increasing exponentially. In fact, I had an interesting discussion with Gordon Moore of Moore's Law, and I argued that digitization of biology was changing faster than Moore's Law. And he agreed, but he said it didn't matter because we were dependent on computing to analyze the information, so Moore's Law always wins. But now writing the genetic code has proceeded very slowly until recently, and it is now in the logarithmic change period. We started back in 1995 asking these questions. We thought it would be simple just to synthesize very short pieces of DNA and then assemble them into small viruses. It turned out to be extremely difficult because the process for synthesizing DNA is a degenerative process where the longer you make the pieces, the more errors. So several years ago, we developed with my team, Ham Smith and Clyde Hutchison, new methods for making the DNA very accurately. And we tried it with a test for a small bacterial virus called Phi-X174. This is only around 5,000 letters of genetic code, and historically it was the first first virus that Fred Sanger decoded the genome of. So after starting with the information in the computer, we made the pieces of DNA, went through the error correction, and 2 weeks later had a piece of DNA that we knew was 100% accurate. We inserted that in bacteria, and the bacteria recognized this DNA as software and began to make the viral particles from the chemical piece of DNA. When the virus was released from the cells, it actually killed the bacteria, sort of making true the fable about ingratitude and killing your host. It happens all the time in biology. But we think this was very exciting because it was clearly a piece of software that led to the construction of its own hardware. In this case, the hardware is a virus that encapsulates the DNA and can kill bacterial cells with it. Our plan on making a bacterial chromosome was then based on making viral-sized pieces of DNA and trying to put them together. DNA, as it gets larger, becomes very brittle and very hard to work with. Nobody's ever made very large pieces, so we had to, over the years, develop a number of new techniques. One of the things as we think of designing DNA from the digital world, you can only design things as accurately as the information in the digital world. And unfortunately, most of the data in the public databases is not of sufficient accuracy to regenerate life from. In 1995, when we sequenced the mycoplasma genome, the standard in the community was 1 error per 10,000 base pairs, which for reading the genetic code was considered satisfactory. but if you're going to write the genetic code, it's not acceptable. So we resequenced the genome. In fact, we found 30 errors that would have rendered this piece of DNA inactive. But in designing the DNA, you have to start with the large molecule, design pieces 50 to 80 letters long that go together to make subunits that then have to be designed so they go together. In addition, we use the genetic code. So the genetic code has 4 letters, A, C, G, and T. And sets of 3 of those letters code for our 20 amino acids. Each of those amino acids have a single letter designation. So you can actually use the genetic code as a means of writing information non-related to genetics. So we use this for watermarking the genome. It would have been easy to write India today in the genetic code using this information so that we could always prove this was a man-made chemical, not a natural contaminant. So how do you put all these pieces together? Well, trying several methods over the years, we found there's natural biological processes that do this called homologous recombination. In fact, there's organisms that look like they came from other planets, such as Deinococcus radiodurans. That can take 3 million rads of radiation, a tiny fraction of which would kill us. Its chromosome gets blown into little pieces, but over 12 to 24 hours it reassembles its genetic code precisely the way it was before, and the organism can start replicating again. So this is one of several organisms that could easily survive in outer space, even with minor shielding in a rock or a meteor, could last perhaps millions of years absorbing ionizing radiation. We use these recombination effects to build the chromosome. This is actually the first photo taken of it. It's such a large molecule that we only need a light microscope. We don't need an electron microscope. It's a circular piece of DNA, so you can see the circle moving over a 6-second time period. And this was our publication that you may have heard about, because this is the largest man-made molecule by a substantial margin. It's over 300 million molecular weight. It takes over 140 pages just printing one letter at a time at a font of 10 with no spacing to print out the 582,000 letters of genetic code that we have. So the next big challenge is how do we boot up a bacterial chromosome? And that's still in process with this synthetic molecule. But last year we published what I think is maybe one of the most important papers that we've done, because it helps us understand evolution in a way never been studied before. And that was transplanting a genome from one organism to another, completely changing one species into another. So we isolated the DNA from one microbial species. We added enzymes that chewed up all the proteins with this, so it was just naked DNA. We added a few extra genes to it so it would be easy to see. It turns the cells blue and also allowed for selection. And we put this DNA into another related microbial cell without removing the chromosome, but we used some unique tricks of microbiology and molecular biology. And I'm sure you'll appreciate this very advanced graphic that we have. So we inserted the new chromosome into the cell, but it turns out the chromosome that we inserted codes for the restriction enzymes, the molecular scissors that cut DNA. The chromosome in the cell did not have any of those genes. So as soon as the cell began to read the inserted chromosome, it expressed these enzymes, which then recognized the original chromosome as foreign and destroying it. A short while later, the cell, all the characteristics of the cell of the original species disappeared, and it converted entirely into this new cell, this new species. You can see that it's blue. All the proteins now are those just coded for by the transplanted chromosome. Now this may sound like science fiction fantasy, but in fact it turns out this is a key part of how evolution has proceeded. A lot of anti-evolutionists argued that just simply making point mutations, we could never get to complex structures. But when we look in the genomes of species around the world, for example, the cholera genome, many people thought the cholera genome would just be like E. coli with a single chromosome. It turns out cholera has two chromosomes, one from one organism and one from another, and bestows these new characteristics to it. So if you transplant an entire chromosome, instead of changing one letter of the genetic code, you're literally adding thousands of new features in an instant. And imagine this happening over and over again throughout the 3.5 billion years of evolution. So what are the next steps and why? Obviously, we're trying to boot up the synthetic chromosome, but we've already gone on to the next stages of trying to use these new tools to solve some of the major problems facing all of us. When we look at the biggest problems that I see on the planet, it's what we're doing to our environment. It's the 4.2 billion tons of CO2 we add net to our atmosphere each year, primarily from burning oil and coal. And so we need new replacements. People have been looking at feedstocks from agriculture. Those experiments in the U.S. of corn to ethanol are failing pretty badly. We're trying a number of new approaches, but biology can be done on incredible scales. Just through engineering microbial cells, DuPont now converts a 6-carbon sugar into the 3-carbon propane diol using these giant fermenters. They're 600,000-liter fermenters. They literally grow up tons of this metabolically altered, genetically altered bacteria to do the chemistry that is far faster and cheaper than chemists trying to do the same work. We have a program with BP trying to see if we can convert coal into natural gas using natural organisms. We've taken samples, as we did from the ocean, only this time a mile deep in the earth. And we found more diversity, more organisms, more biodensity a mile below us than we found in the oceans. These organisms actually will live off of coal. I don't know if you can see this electron micrograph. This is the bacteria on the surface of the coal. It actually eats the coal and digests it, converts it into natural gas. In fact, that's why there's explosions in coal mines due to these natural processes. We're seeing if we can change them, even a 1% change would have a huge impact on the natural gas industry. The same organisms, if you can see in the one test tube, produce bubbles of methane, of natural gas, just by digesting the coal. While this is still using carbon out of the ground, it's about a 10-fold improvement over mining coal and burning it, because we don't have to mine it. We can convert it in the earth to natural gas using natural processes. The long-term future is through designer fuels. We have second and third generation fuels. If ethanol is the first generation, that are far superior to ethanol, that can be made starting with sugar as the feedstock. But sugar as a feedstock is going to be very limiting, and so we're working in new directions. So with these 20 million genes that we've discovered, we're viewing these as the design components of the future. We have software we can actually design metabolic pathways in organisms to produce fuels, ideally starting back with carbon dioxide. We have new tools in a field we're calling combinatorial genomics, where instead of taking 10 years and $100 million like DuPont did, we can optimize these pathways very quickly just with small teams, screening for chemical production, hopefully change in the chemical industry or fuel production. We're working on our 4th generation fuel that starts with CO2 as the feedstock. It was interesting in the film clip from 2003, you see we've been discussing these ideas for a long time. We now have the ability to start to make these organisms by modifying existing ones first. With CO2 as a feedstock, there's in fact not yet high enough concentrations in most atmospheric conditions for utilizing the CO2. But as CO2 sequestration gets improved efficiency, instead of burying that CO2 deep in the earth, we can just take, we think, pipelines of CO2, convert it back into natural gas or other fuels, and recycle that carbon dioxide instead of just trying to hide it away in the earth. We also have, aside from photosynthesis, numerous pathways that we've discovered from organisms in the environment. This was from the first archaea genome that we published in 1996. This organism lives in near boiling water temperatures. It converts CO2 into methane using only molecular hydrogen as its energy source. Our team at the Institute has also designed microbial fuel cells, and we think these show incredible promise, particularly for developing countries. We're just using bacteria already existing in human and animal waste, putting these devices in. They naturally collinate the anode and cathode. Here's a picture of them using these new nanowires that our team discovered that actually transmit electricity. So we can use the human waste either to make clean, drinkable water, or we can generate electricity with it. It's easy to dial up and choose which direction. So we think there's potential for this new field of synthetic genomics, starting with the digital world, using all these naturally occurring organisms and pathways. We can capture energy from sunlight, we can capture energy from biomass, but more importantly, we think we can recycle carbon dioxide and at least help get to steady state levels, if not reduce some of the contamination we've done to our environment. And these have potential in every area of science and industry, everything from understanding basic mechanisms of life to hopefully starting down the direction that DuPont and others have of stopping to use oil as their raw material, replacing the petrochemical industry with biology, hopefully becoming a major source of energy, bioremediation, and even working, we have a project on making combinatorial chemicals for new antibiotics and vaccines. This area has raised a lot of discussion as we as a species can start in this digital world and design biology, even though humans have been modifying biology ever since there was any kind of cooperative society. Every plant we grow has been extensively modified over centuries and centuries. But before we did the first experiment, we started with a bioethical review. The results of that were published in Science by that team in 1999. The Sloan Foundation has funded us along with MIT to do a complete review. There's a new part of the executive branch of the U.S. government looking at dual use research and tracking the positive and negative benefits of this exciting new technology. Thank you very much.